So, Assalamu alaikum, everybody. I'm Rula Ismaiel. I'm a second-year medical student, and I'll be teaching you Glycolysis 1 and 2. Hopefully, this spa session won't last more than an hour, but obviously, things do happen along the way. So, shall we start? Okay. Wait. Okay. Table of contents. You have some background info that was mentioned in your slides. You have the overall pathway and the reactions Dr. Hannah mentioned, glycolysis regulation, clinicals, the fate of pyruvate and enzymes overview, and a few questions. Okay, so why do we need glycolysis? We need glycolysis to generate ATP. ATP is the energy currency that is used throughout the body. It's generated by the oxidation of sugars, amino acids, fatty acids, and to use that energy that ATP has, you need to hydrolyze it, okay? And what I mean by hydrolyzing it is breaking the bonds. The bonds is where the energy needed for the synthetic reaction, synthetic reactions to carry on are present, okay? And you can see over here, can you guys see my cursor or do you want me to use a pointer? Turn on laser pointer. Okay, there we go. So you see these bonds over here, okay? These are high energy phosphate bonds. And when they're broken down, hydrolyzed, they're the ones that release the energy. When ATP is hydrolyzed, it becomes either ADP, which is diphosphate, and ANP, which is monophosphate. Okay, so basically, one phosphate is lost, two phosphates is lost. There's GTP, UTP, CTP, and other, currencies being used, but it's mainly ATP. Now, ATP is used for the sodium potassium pump, and without ATP, these pumps fail, which causes And without ATP, these pumps fail, which causes an disequilibrium in the cell membrane, causing it to lysis or rupture. Okay, so that's the background for ATP. Let's see. Okay, now how does glucose enter the cell? Please, this is such an important concept, specifically the GLUT2 and GLUT4. They've been asked on multiple occasions, and this is very high yield. I'm sorry, it's very memorization heavy, but it's also very, very simple. Glucose enters, and then glucose enters from one way and then goes out the other way into the cytosol. GLUT1, it's found everywhere. GLUT2, it's the liver, intestines, pancreatic, B cells, beta cells. Beta cells, they release the insulin needed to lower blood sugar. Okay, then you have GLUT3, which is brain. You will cover GLUT3 and brain glucose usage later on in MOL 2, but for now, just know, like in the back of your head, the brain uses GLUT3, and GLUT4, it's for muscle, adipose tissue, and the heart. Dr. Hannah loves asking about muscle and adipose tissue, so always keep that in mind. Now, GLUT4, the thing that makes it special in a way is that it's insulin dependent, which basically means if there is insulin, it will open, okay, to lower the blood sugar, okay? If there's no insulin, it won't open. There's no need to lower blood sugar because you still need to have a baseline level of glucose in the blood, okay? So that's the concept for this one. Again, very sorry, but just focus on these and the fact that this one is insulin-dependent glucose, okay? Now, keep in mind, glucose is for transport, okay? And glycogen is for storage. Glycogen is stored in the liver and the muscles, but mainly the liver, okay? After 1 day of fasting, if you've been fasting for a whole day, the liver starts to make glucose by gluconeogenesis. So it's basically synthesizing glucose to maintain a glucose level in the blood from glycogens, amino acids, or fatty acids, okay? That's it for this slide. Then you have Acetyle-CoA. Acetyle-CoA oxidation of glucose is what we're focusing on for this past session specifically. 2 carbons of acetyl group are oxidized and it makes carbon dioxide, okay? 2 carbon dioxide, sorry. The excess hydrogen is picked up by NAD plus and FAD2 plus, giving NHD and FADH. They're basically intermediates. They're kind of like the shuttle bus for the extra electrons that are produced by metabolic processes like glycolysis, the Krebs cycle. You'll learn more about the Krebs cycle in other past sessions, but for this one, I'll just go I'll just touch it, okay? And NADH and FADH2, it's used in the electron transport chain in the mitochondria, which is this basically. They just pick up the electrons early on, and then they give it to the mitochondria. The mitochondria does its thing and then makes ATP. Electron transport chain is where a lot of ATP is produced. In glycolysis, it's just a little bit, okay? Now, keep in mind, acetyl-CoA, you don't need to know the structure. I just wanted to show you what a thioester bond is for the sake of completion, because it was mentioned in your lecture, but it wasn't tested on in past papers, okay? So this is a thioester bond. These are the 2 carbons over here that are being used to oxidize and make carbon dioxide, okay? That's it. Now, is everyone okay so far? Do I move on? I'm going to take that as a yes and move on. Now, this is the overall pathway of glycolysis, okay? I really, really, really don't like the diagram that Dr. Hannah provided. Yeah, I really don't like the diagram Dr. Hannah provided, so I just pulled this one online from the internet. Now, you need to focus on reaction 1, 3, and 10, but that doesn't mean you skip out on the other reactions that were mentioned in the lecture, okay? You have glucose, glucose 6-phosphate, bunch of intermediates. I'll be going back to this slide a lot just to show you, but yeah, that's it. Now, reaction 1 is glucose becoming glucose 6-phosphate with the help of hexokinase. Now, keep in mind, whenever you see a kinase in the name of an enzyme, that means it's using a phosphate and putting it into the molecule, okay? So you can see here, ATP is becoming ADP. That means 1 phosphate is taken out of that ATP and it's put into glucose, which makes it glucose 6-phosphate. And now, it's an irreversible reaction. Keep that in mind. And keep in mind that any phosphorylated intermediate can't leave the cell. There are no special carriers for it. So yeah, that's all you need to know. Another thing to point out is that the liver and the pancreas, they do not use hexokinase, they use glucokinase, okay? The liver, first of all, doesn't use glucose as its source of energy. Glucokinase phosphorylates glucose, yes, but only to reduce hyperglycemia. It's basically just to take it, just to take it out of the blood and lower hyperglycemia. It's induced by insulin for storage of glucose. Okay, glucokinase is induced by insulin for storage of glucose. Also, another thing to keep in mind, I will be going all over the enzymes towards the end of the slides. Okay, the pancreas also uses glucokinase to sense the levels of glucose in the bloodstream, so it can release insulin from beta cells to regulate it. So glucokinase in the pancreas is acting as like an antenna. Okay, I sense that there's a lot of glucose in the blood, so I need to release insulin so it can be lowered. Because if you have constant a very constant high blood glucose, that's really bad for your cells, and you don't want that. So that's what the pancreas does. Okay. Now, this is a very important yet very difficult topic. Please let me explain it. Okay, and I really ask for some of you to be interactive with me for this one. And if you have questions, just wait until I'm done with the slide and then you can ask. Okay. We're gonna focus on this graph over here. Okay, I'm gonna ask, does anybody know what Km is? And if someone can answer voicely, like with the voice chat, that would be amazing. Guys, please. Okay. No one's answering. Yes? No?
Affinity.
It's related to affinity, yes. It's the Km is a measurement of the how do I explain it? Like, Km is a measurement of when half of the binding sites available on the enzyme are occupied. So I have, let's just say, 100 enzymes. Okay, I have 100 enzymes. 50% of these are occupied. 50% of the binding sites have a substrate that they're working on, trying to break up or trying to synthesize. Okay, that's Km. The way Km is related to affinity, it's related in the sense that it's inversely proportional to affinity, which basically means if Km is low, that means affinity is high. Okay, and if Km is high, affinity is low. Does that make sense? I'm going to take that as a yes. Okay. So here you can see the graphs of glucokinase and hexokinase. For affinity, okay, it can be deduced from the graph by the shape of the slope, by how steep the slope is. Okay, so if I have a very steep slope, that means it's has a high affinity for whatever substrate the enzyme is working on. So like in the case of hexokinase, okay, it has a very steep slope, so that makes it high affinity. Glucokinase, it has less steep of a slope, so it makes it a lower affinity. Okay, now Vmax is when the graph plateaus. So all the active sites on the enzymes are filled. They're busy working, they're doing something. Okay, so that's when the graph literally plateaus, it goes flat. Okay, and hexokinase reaches the Vmax much quicker, like it plateaus much quicker compared to glucokinase, for the whole reason that hexokinase has a higher affinity for glucose compared to glucokinase. Okay, am I making sense right now? Is everything okay? Guys, I would really like someone to tell me if everything's okay. Okay. Now, this is just stuff from your slides. I'll go through them very quickly, and if you have questions, let me know. Okay, so all tissues, hexokinase is found in all tissues. Lower Km, high affinity for glucose. It permits efficient metabolism of glucose even when tissue concentrations of glucose are low. So whether there's high glucose or low glucose, it's still working. Okay, Vmax is low, cannot phosphorylate more sugars than the cell can use. Okay, so there is no point of a cell to keep phosphorylating sugars when the cell isn't using it. Okay, that would be a waste of energy. That would be a waste of ATP rather than producing ATP, which is not the case. We don't want to do that. Okay, and most importantly, it's inhibited by glucose 6-phosphate. What usually happens, or at least the way I like to visualize it, is that okay, there's a lot of glucose 6-phosphate. Okay, it tells the enzyme, tells hexokinase, hey, we have enough. There's no need for more. Please stop. Okay, so that's how it's inhibited. Okay, liver and pancreas, that's glucokinase, increase Km, decrease affinity for glucose. It only functions when the intrahepatic glucose concentration is high. Okay, increase Vmax, allowing the liver to effectively remove glucose, minimizing hyperglycemia. Okay, so it's basically working very quickly. It's kind of like all hands on deck type of situation just to decrease blood glucose. During the absorptive period, basically after you eat your food, by a few minutes, you start absorbing everything. That's the absorptive period. And it's activated by insulin. High K, someone's asking, high Km means most of the enzymes are busy with high sugar. Okay, let me show you on the graph. Let's take the case of glucokinase. Enzyme activity, there's a lot of sugar. Okay, let's just say there's 20. Okay, goes all the way up. Okay, that means enzyme activity is really, really high. Does that make sense? So in that case, I believe yes, most of the enzymes are busy with high sugar. But it has a low affinity. So the affinity is like the substrate binding to the enzyme. Okay. So Km is how busy the enzyme is. I wouldn't say so. It's more of like half of the active sites of an enzyme are occupied by the substrate. I have the definition here. How Km, so Km is how busy the enzyme is. And then to an extent, I feel so, yes. Does anybody have more questions before we move on? Concentration, no, it's not the concentration of substrates attached to an enzyme, it's the active sites that are occupied by the substrate. Okay, so as I was saying, 100 enzymes, okay, 50 of them are occupied with something. That's Km, okay? And you can deduce Km by going to Vmax, okay? By going to Vmax and then doing 50, oop, going down. So that's 10 Km is the Km. Does that make sense? Am I good to move? Okay. You have reaction 2, which is the phosphofructokinase. I'm going to refer to it as PFK1 from now on. Fructose 6-phosphate becomes phosphofructokinase because, fructose 6-phosphate becomes fructose 1,6-bisphosphate with the help of phosphofructokinase. Okay, bunch of fructose, bunch of phosphate, doesn't matter. Point is, you need to know that PFK adds another phosphate, okay, with the help of ATP. So far we have used 2 ATPs in glycolysis, and the whole point of glycolysis is to make ATP, okay? But we've already used, so it doesn't make sense right now, but will make sense later on, okay? Now, it's both the rate limiting step and the committed step. Can someone please tell me what's the difference between both of them? Type it on the chat if you want. It's okay. I'm checking it regularly. Okay. Rate limiting, the slowest part. Yeah, you're almost there, Mariam. And dictates the other reactions. Exactly. Rate limiting, okay, is kind of the drive of the reaction. So it controls whether it goes very fast or very slow. Okay. If the reaction goes in one direction, it means it will lead the direction. It will read the reaction. Yes, exactly. So the committed step, once that step has been passed, the intermediates, they have nowhere else to go. They keep following that specific path. Okay. So like fructose 6-phosphate, it can be used elsewhere. Glucose 6-phosphate, it can also be used elsewhere. But fructose 1,6-bisphosphate is the committed step. So when fructose 1,6-bisphosphate is made, it doesn't go anywhere. It just keeps going on with glycolysis. Does that make sense? I take that as yes. Okay. PFK1 is activated by AMP, fructose 2,6-bisphosphate. I'll explain this in a second. And it's inhibited by ATP and citrate. Fructose 2,6-bisphosphate can override any inhibition in liver cells. PFK1 in liver, okay. Sorry. First off, this doesn't make sense to me. I'm a second year and it still didn't make sense to me. This diagram never made sense to me, but I kind of summarized it in this. I hope it makes much more sense. Yeah, okay. So in the well-fed state, insulin is released. When insulin is released, it's stimulating PFK2. Okay, PFK2 is found in the liver. It's not found anywhere. So when PFK2 is stimulated, it catalyzes the reaction of fructose 6-phosphate to fructose 2,6-bisphosphate, okay? Which later on, this intermediate, fructose 2,6-bisphosphate, activates PFK1, okay? Please keep in mind the different, like 2 and 1 and what they do. PFK1 then stimulates fructose 6-phosphate to fructose 1,6-bisphosphate, so it can continue with glycolysis. And it's dephosphorylated to make it active. Glucagon, on the other hand, it kind of stops all of this reaction. It decreases fructose 2,6-bisphosphate, so it lowers kind of, how do I explain this? Kind of lowers the activity of PFK1, so it doesn't create fructose 1,6-bisphosphate. Does this make sense to everybody? Okay, I'm gonna move on. Now you have reaction 6, which is glyceraldehyde 3-phosphate dehydrogenase. You don't need to know the name of the enzyme, I believe, for this one, but keep it in the back of your head. The whole point of this slide is just know that this specific reaction in glycolysis requires NAD+. Okay, that's literally it. This did come up in past questions, so if you're gonna go home with anything from this past session, just remember reaction 6 requires NAD+. That's it. Okay, there are two high energy intermediates, 1,3-biphosphoglycerate and phosphophenol pyruvate. Okay, and you can see here in the reaction, okay, how NAD+ is being used. Okay, and dehydrogenase. Okay, so you're removing a hydrogen from a molecule. Okay, and giving it to NAD+, so it can carry it away to the mitochondria for the electron transport chain. Okay. What else I got? Okay, now this is the checkpoint. Does anybody have any questions? I take that as no. Okay, I'm gonna explain another concept on this checkpoint, which is fructose 1,6-bisphosphate. Fructose 1,6-bisphosphate is split into two intermediates. Dihydroxyacetone phosphate. You don't need to know these intermediates, but I'm trying to deliver a concept here, which is that it splits into two, 3 carbons here, 3 carbons here. Okay. And the other one is glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate will continue normally because there is an enzyme for it. However, this one, dihydroxyacetone phosphate, there isn't. So it won't make sense for it to just exist in the cell and accumulate and cause toxic side effects. Instead, it will be isomerized, and an isomerase just basically changes the arrangement of a molecule. Okay, and it becomes glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate becomes 1,3-biphosphogycerate. Now, since the molecule is being split into two, and then we're making 2-glyceraldehyde 3-phosphate, it will continue with the reaction normally. Okay, so everything from reaction 7 onwards is multiplied by 2. Okay, so 2 NAD pluses are used, 2 ATPs are being made, 2 ATPs are being made here, and 2 pyruvates are being made as well. Okay, because this one splits. Okay, and then this one changes, and then it goes back, it goes normally, continues the normal flow. Okay. Reaction 7 is a substrate level phosphorylation. It's the first step in glycolysis to make ATP. Okay. And it takes the phosphate, it takes this phosphate over here specifically. Again, you don't need to know the structure of the phosphate of the molecule, sorry, you just need to know the concept. Okay, it takes this phosphate, puts it in an ADP to make ATP, and it makes out 3-phosphoglycerate. Okay, now keep in mind 3-phosphoglycerate, you kind of need to change it up a bit, and you need to make it into 2-phosphoglycerate by an enzyme called phosphoglycerate mutase. Now, mutase is just an old way of saying isomerase. Okay, so this isomerase over here, okay, olden days it was also called mutase, but now it's called isomerase. Now, for some reason, they didn't change this one. I don't know. It's not up to me. I'm not a biochemist. I'm a medical student. From reaction 7 onwards, everything is multiplied by 2. So 2 ATPs are being produced in this specific reaction. Okay. Reaction 9 is enolase. Okay. And it's a dehydration reaction. Please keep in mind the difference between dehydrogenation and dehydration. Dehydration removes water. You're dehydrating something. You're taking out a water molecule, which is H2O. Dehydrogenation is the fact that you're taking out a hydrogen. Okay, so that's the difference in the naming. Okay, and this carries on with all the other enzymes, by the way. It's not specific to glycolysis. Now, reaction 9, enolase, 2 phosphoglycerate becomes PEP, phosphophenol pyruvate, with the help of enolase. The main takeaway about this slide is that it's inhibited by fluoride. Okay, and fluoride, where is fluoride used? It's used in toothpaste, and it helps in the prevention of caries by strep mutants. You don't need to know about strep mutants, but just know it's inhibited by fluoride, and it prevents caries. Now, the whole mechanism behind this is that bacteria has enolase. It's if you don't brush your teeth, there's still like sugar in your mouth and everything. So the bacteria is eating it up, breaking down the sugars in the oral cavity, and there would be a very high accumulation of acid, okay? And that breaks away the enamel of your teeth. And when that happens, you kind of get caries. So you inhibit enolase in bacteria, okay, in the oral cavity. Obviously, the fluoride is not being absorbed in your blood. So you inhibit the enolase in the body, in the bacteria, sorry. And yeah, the bacteria can't break down sugar. It can cause caries. That's all the takeaway that you need to know. I don't think you need to know the mechanism, but just know it's used in toothpaste inhibits this enzyme. Okay. Now, pyruvate kinase, we're at the last step, okay, before entering, or actually before the last step, before entering pyruvate, the Krebs cycle. Now, PEP, okay, gets converted to pyruvate with the help of pyruvate kinase. And this is the second substrate level phosphorylation, and it's the third regulated step. Now, reactions 1, 3, and 10, okay, these are the 3 regulated steps. Okay, we spoke about 1, we spoke about 3, and now this is 10. This is like the final one. Okay, so PEP becomes pyruvate with the help of pyruvate kinase. It makes ATP. Now, is it 1 ATP or 2 ATP? It's 2 ATP. Okay, and how many pyruvates are being produced? Same thing, 2 pyruvates. Okay. Something to stay on the lookout for is fructose 1,6-bisphosphate. Fructose 1,6-bisphosphate is a feed-forward activation, which basically means an earlier intermediate of the cycle, okay, or the reaction, kind of stimulates the enzyme that's towards the end, okay, and tells it, hey, kind of get ready, we're there, we're coming, just, be on your tippy toes so that you can create the final product. Instead of just like all of a sudden you have a lot of PEP and all the enzymes are occupied and they're like, whoa, what is going on? Okay, that's how I like to think about it. Okay, and obviously pyruvate kinase is inhibited by ATP, because if there is ATP, there's no point for this enzyme as well, to make more pyruvate, which can be oxidized later. Okay. Now, some completing information, okay, is that glycolysis occurs in the cytoplasm, okay? And RBCs rely on glycolysis for ATP. Why? Because they don't have mitochondria. And a red blood cell is biconcave. Like, guys, you just finished CVP. There is no mitochondria in the RBCs, okay? Glycolysis does not require oxygen. In any of the reactions I've just mentioned, was oxygen involved in this? No, obviously. Now, there's net production of 2 ATP and 2 NADH. And glycolysis needs NAD+. No NAD+, no glycolysis. Where's NAD+, being used as? Which reaction? Guys, please someone use the chat. Okay, I'm gonna answer. You guys are taking too slow. It's reaction 6. NAD+ is used in reaction 6. Okay, now you can see here, this is reaction 1, this is reaction 3, and so on and so forth. 2 ATP, we invested 2 ATP, we gave glycolysis 2 ATP, and it's giving us 2 ATP back. Okay, and these 2 ATPs, okay, is what's being used by RBCs to carry out their normal function. okay, as well as 2 NADHs from um, reaction 6, okay? Glycolysis regulation, okay, in the well-fed state and the starved state. Well-fed, you just had a meal, you're good, and starved when you haven't had anything. Now, well-fed, decrease glucagon, increase insulin, okay? And that increases fructose to 6-bisphosphate. It all goes back to the earlier slides I just mentioned. Remember how I said, oh, insulin activates PFK2 to make fructose to 6-bisphosphate? That's how you kind of link things together, okay? Now, since there is an increase in fructose 2,6-bisphosphate, there is an increase in glycolysis, okay? Because fructose 2,6-bisphosphate is pushing PFK1 to catalyze the reaction needed to make the intermediate step, the rate limiting step, and the committed step as well. Okay, so when fructose 2,6-bisphosphate is increased, there's an increase in glycolysis because it affects PFK1. Increase and decrease in gluconeogenesis. Starved is complete opposite. Increased glucagon, increased insulin, decreased fructose 2,6-bisphosphate, and decreased glycolysis and increased gluconeogenesis. You still need glucose in your body, but your body's like, okay, there's no free glucose from the outside sources, food. Okay, so let me use what I have in storage. Okay, now insulin and glucagon work on enzymes in reactions 1, 3, and 10. Reaction 1, glucose becomes glucose 6-phosphate with glucokinase. Reaction 3, fructose 6-phosphate becomes fructose 1,6-bisphosphate with PFK1. Reaction 10, PEP becomes pyruvate with pyruvate kinase. I just mentioned these are just like repetition. The more you repeat the lecture, the more it sticks in your head. Okay, now clinicals. It's only these 2 clinicals that were mentioned, I believe, and lactic acidosis, but that's another thing. You need to know that in pyruvate kinase deficiency, there is a backup of PEP. And when that happens, it goes back to 1,3-biphosphoglycerate. And 1,3-biphosphoglycerate, when it accumulates, it becomes 2,3-PBG. Okay, PBG, it's used for this graph. Do you guys remember this graph? The oxygen affinity graph of hemoglobin from Foundation? I'm praying that you do. Okay, and it was taught in CVP as well. Okay, so this increases, this graph, this is the normal one, it shifts to the right. And what that means is that the oxygen affinity decreases. Okay, so when that happens, there's not a lot of oxygens being passed around to organs, cells, muscles, whatever it is. Now, keep in mind that pyruvate kinase deficiency is very different, is kind of the same, but at the same time, quite kind of different to G6PD deficiency, in the sense that G6PD has Heinz body, but pyruvate kinase doesn't have Heinz body. Okay, and you can tell over here, like you see those little dots, those are Heinz bodies. Pyruvate kinase deficiency doesn't have Heinz bodies. That's how you differentiate between them. Okay, you'll also see hemolytic anemia. This is one of the causes of hemolytic anemia. However, this is not the main cause of hemolytic anemia. It can be caused by a lot of things, but this is one of them. Okay, so that's for pyruvate kinase deficiency. Also, the whole reason that RBCs swell and they rupture is because the sodium potassium pumps are not being maintained. Okay, so the equilibrium it becomes weird. Okay, now Moody 2 is maturity onset diabetes of the young. This, I've never seen a question on it, but keep it in the back of your head. Glucokinase is used to measure blood glucose and release insulin accordingly. We know this, it's in the pancreas. Okay, so if anything is wrong or mutated in the monogene, the autosomal form of diabetes, it causes diabetes in the young. Okay, so you would see like someone 13, 14, maybe even younger with diabetes, which wouldn't make sense because that person is healthy unless they're extremely obese. But in this case, it's no, it's just that the receptor is not working. It can't sense the levels of glucose in the blood. Okay. Is everything okay so far? Do I need to go back and review anything?
It's good.
It's good. Okay, thank you. Now, what happens to pyruvate? Pyruvate in aerobic conditions, aerobic means in the presence of oxygen, becomes acetyl-CoA. It gets oxidized specifically to become acetyl-CoA. Now, in anaerobic, okay, or without oxygen conditions, it becomes either ethanol or lactic acid. Now, ethanol is in yeast or bacteria, okay, but lactic acid is in RBCs and muscle cells. Pyruvate gets reduced, pyruvate gets oxidized. Oxidation is for acetyl-CoA, reduced is for ethanol or lactic acid. Okay, so you need to know for this bit. Okay, anaerobic glycolysis. Okay, pyruvate becomes lactic acid. Okay, let me talk about this for a second. Pyruvate becomes lactate with the help of lactate dehydrogenase. There's NAD plus being produced. When NAD plus is being produced, it's being used in the glycolysis cycle, and the reaction that uses glycolysis that uses NAD plus is reaction 6, obviously. Now, it's used during mature RBCs for the production of ATP. Again, no mitochondria. ATP is not being produced by the mitochondria, but we still need a supply of NAD plus. Okay, so this is a reaction in RBCs specifically that helps with like the proliferation of NAD plus, keeping the supply of NAD plus. Skeletal muscles during high intensity exercises, since oxygen becomes a limiting factor. Basically, the supply can't meet up with the demand. Okay. And ischemic tissues for crisis management will eventually lead to cell death due to accumulating lactic acid. Now, ischemia is basically part of a bunch of cells. They're not receiving blood. There's a block of the blood vessels, or there are no blood vessels, or there's no RBCs reaching them whatsoever. Okay, so they begin to like die out. Okay, or rather, they become ischemic. They basically get injured. Okay, so in the time, there's like a specific time frame where the ischemic tissues, they can still recover back to normal. In that period, they will use anaerobic glycolysis to, they will use anaerobic glycolysis to kind of keep up with cell functions, with basic baseline minimal cell functions. If it's still continuous ischemia, it becomes necrosis, okay? Because of accumulating lactic acid levels. Anaerobic glycolysis regenerates NAD plus needed for glycolysis, okay? That's it. This is really important. And accumulation of lactic acid will result in acidosis. You guys have taken CVP, you just finished with it. I'm sorry to traumatize you again, but you guys know what acidosis is at this point. Fingers crossed. Okay. Without oxygen, the mitochondria cannot oxidize NADH to NAD plus, and we said NAD plus is important, which pushes the reaction in direction of lactate formation. Okay. So NADH, if there's a lot of it, it pushes the reaction. It shifts the equilibrium to make lactate rather than, pushing it back to pyruvate. Okay. Lactic acidosis. Yes. What about in the muscles? Isn't there also lactate formation during exercise? There is. However, with the exercise, you do eventually end up taking a break. You're like, okay, I'm really tired. Let me catch my breath. So the blood can reach those muscles, and it kind of reverses the how did I say this? The acidosis that is being caused by the lactic acid accumulation. Does that make sense? Yes? Does that make sense to everyone? Okay. Lactic acidosis, due to impairment of oxidative metabolism by respiratory failure, insufficient oxygen transport, or toxins that prevent oxidative phosphorylation. Okay? Fancy words basically telling you either the lungs are not failing, there is not enough oxygen reaching, or there are some toxins that are stopping the Krebs cycle or the citric acid cycle. Okay, so basically the cycles can't keep going to make the ATP needed. Okay, tissue ischemia, PFK1, is stimulated by low energy. Therefore, large amounts of lactate are formed by anaerobic glycolysis. Okay, so PFK1 we mentioned before. Okay, it's making fructose fructose 1,6-bisphosphate. Now, PFK1 realizes that there's little to no energy, so it's like, okay, I need to make energy. I'm the one who's taking up all the work. I'm the important one. Let me make more energy. Let me make more ATP. Let me push glycolysis in the fate of pyruvate. However, pyruvate gets made, okay, but it doesn't go to the Krebs cycle, okay. Instead, it goes to the formation of lactate. Does that make sense? Yes, please. Okay. Now this, okay, you guys have covered stimulated, inhibit, and the notes. But you need to know if an enzyme is active when it's phosphorylated or dephosphorylated. Okay, in hexokinase, it's not affected. In glucokinase, it's also not affected. PFK1, it's also not affected. PFK2, when it's dephosphorylated, so you took out the phosphate from the enzyme, it's on. Pyruvate kinase, when it's also dephosphorylated, so you took out the phosphate, it's also on. So it makes it active, okay? It pushes the reaction to the intermediate that it needs to make. Now, hexokinase is not stimulated by anything per se. Glucokinase, it's stimulated by insulin. We've gone through this. PFK1, it's stimulated by AMP, low energy, okay? And fructose 2,6-bisphosphate, okay? Again, mentioned this before. PFK2, sorry, is stimulated by insulin, and pyruvate kinase as well, insulin. Hexokinase inhibited by glucose 6-phosphate. PFK1, ATP and citrate, it's inhibited. Everything else glucagon. And here are some notes. These were mentioned earlier in the lecture, but I feel like I'm blabbering too much, so there's no need for me to go over them. But if you have questions, please let me know. The conditions in the previous slide. Okay. I believe you need to know physical exercise, high altitude. I mean, most of these, I believe you guys have taken, right? But I would like to say no. But physical exercise, high altitude, drowning, I'd like to say no, but you guys already took this, I believe. You guys already covered this. Yes or no? At least a big chunk of them. Dr. Hannah didn't mention anything. So, okay. One GERD disease, I think no. Pyruvate dehydrogenase deficiency, I think as well, no. But leukemia metastatic carcinoma, you guys haven't taken these. Just keep in mind from alcohol intoxication up. Keep them in mind. I'm not saying memorize them. I'm just saying keep them in mind. Okay. What's next? Okay, questions. I'm really hoping I can get more than one answer. Please, more than one person answering these. So which one of the following is a regulatory mechanism employed by muscles for glycolysis? A. Inhibition of PFK1 by AMP. Inhibition of hexokinase by its product. Activation of pyruvate kinase when glucagon levels are elevated. Inhibition of aldolase by fructose 1,6-bisphosphate. And inhibition of glucokinase by fructose 2,6-phosphate. Okay, which one do you guys think it is? Okay, can I have someone else other than Mariam answering, please? Guys? Okay. Mariam, you are right. It is B because inhibition of hexokinase by its product The product is glucose 6-phosphate. It's glucose becoming glucose 6-phosphate with the help of hexokinase. As I mentioned earlier, glucose 6-phosphate is like, enough, we have too many of you, there's no need, okay? That's it for question 1. Question 2 is a patient with type 1 diabetes self-injected insulin prior to their evening meal, but then was distracted and forgot to eat. A few hours later, the individual fainted, and after the paramedics arrived, they did a stat blood glucose level and found it to be 45 mgs over deciliters, I believe. The blood glucose level was so low because which one of the following tissues assimilated most of it under these conditions? This is related back to the GLUT transporters. Okay, so which one would be the answer? Okay, insulin dependent. Which one was insulin dependent? Which GLUT transporter was insulin dependent? And from that GLUT transporter, where is it found? Okay, no one's answering me. Point is, GLUT4 is insulin dependent. This diabetic injected, okay, and it made the GLUT4 channels open up. Glucose entered the cell, okay, and there is hypoglycemia in this case specifically, okay. Now GLUT4 is found on muscles and adipose tissue. Which one are muscles mentioned here? No, it's adipose. Okay, so the adipose tissue took out all of the glucose. Okay. Let's see. Also, the explanations are found in the notes section of the slides for question 1 and question 2. Okay. Now, question 3 is a TBL question that we've done last year. Okay. You are studying the impact of different oxygen levels on erythrocyte cultures. Under low oxygen pressures, a crucial glycolytic pathway intermediate is quickly depleted. Further cell analysis reveals that lactate dehydrogenase activity is significantly lower than expected. Under these hypoxic conditions, which intermediate is most likely going to be rapidly depleted. Okay. Yes, Mariam, it is C. It's NAD+. Okay. Lactate dehydrogenase activity is lower. Okay. So there is no NAD+ being regenerated to be used in the glycolysis pathway. So yes, NAD+ is the one that's being depleted. Okay. Answer C. Okay. Thank you so much, everyone. And I'm very happy to have you here. Please don't forget to fill out the feedback form, and best of luck with MOL 1. You got this, inshallah. And trust me, it's not that difficult. Just try to link everything together.